I welcome you all for today's session. A very good morning to all of you. So I am Dr. Priyanka Sachdev here, and today we are here to continue our live session series. So without wasting any further time, let me start with today's topic. Today's topic is apoptosis in pathology. It's an ultra important topic. So let me start with apoptosis. Okay, so can I start with apoptosis? So let me start with apoptosis. Okay, so first we have understood this diagram in the last session also. I will tell you. So can you see this cell is in homeostasis? In human body, from head to toe, we are made up of cells. Cell is the structural and functional unit of life. In human body, normally all the cells are in homeostasis. What do you mean by homeostasis? That is, the cell is performing their function with a specific structure normally. All the cells in human body are assigned with a particular function. They have a specific structure and a specific function assigned to them. That is homeostasis. But whenever any stress acts on the cell, the stress can be physiological, can be pathological. Three things happen one by one. The first thing, the cell try to cope up with the stress. To cope up with the stress and survive is cell adaptation. If the stress is removed, it's reversible. But if the stress is continued, second thing is happening is reversible cell injury. If the stress is removed now, it's also reversible. But if the stress is continued, finally, the last thing is irreversible cell injury. As the name indicates, it's irreversible. This cannot be reversed because irreversible cell injury is equal to cell death. Actually, once the cell is dead, you cannot make it alive again. So today my topic is cell death. There are two ways the cell dies. There are two ways the cell can be died. It can be apoptosis, it can be necrosis. We can compare apoptosis with suicide and we can compare necrosis with murder. I will tell you why I'm using this simile. Why I'm comparing apoptosis with suicide and necrosis with murder. Now, ultimately, whether it is a suicide or whether it is a murder, the cell is dying. Whenever there is a suicide or murder, the person dies. In the same way, the cell is dying, but the mechanism is different. I will explain you why I'm comparing apoptosis with suicide and necrosis with murder. But for the sake of simplicity, just learn it right now. Okay? For the sake of simplicity, you can learn it right now. Okay? Okay, so let's come ahead. Let me continue with apoptosis. Now, what's apoptosis? I told you in simple language, we can say apoptosis is cell suicide. But what's the actual definition of apoptosis? The first question that may be coming in your mind that, ma'am, why the cell is doing suicide? Nobody wants to do suicide. I do not want to do suicide. Why this cell want to commit suicide? Why? There are two reasons of apoptosis. Either this cell have done its function completely in the body and it is not required in human body. So it will get a signal that you are no more required in human body in this world, you just commit suicide. So that can be the first reason. Okay. The second reason, the DNA of this cell, the DNA of the cell is damaged to such an extent that it cannot be repaired. Whenever there is a DNA damage in the cell, we have DNA repair gene, the mechanics, that is P53. I call them mechanics. So mechanics will try to repair it. But if the damage is beyond repair, we will ask the cell to commit suicide, that you are of no use, your DNA is damaged too much and you should commit suicide. So there are two reasons of apoptosis. Either the cell is not required in human body, its function is already completely performed, or its DNA is damaged completely. Okay, so we ask the cell to commit suicide. So this is the two reasons of the apoptosis I told you. Okay, now how to define? What is the definition of apoptosis in pathology? In pathology language, can you define it? Now looking at the definition, it's a little difficult, right? If I read it, you will say, ma'am, what you are reading, what is this language? Let me explain you the definition with the help of a diagram. Okay, can you see this cell? Now there are two possibilities. Either this cell has done its function completely and it is not required in human body, or the DNA of this cell is damaged too much. It's beyond extent of repair. So we cannot repair it. So we are asking the cell to commit suicide. Ultimately, we destined this cell to commit suicide. Now, you can see in the cytoplasm of the cell, there is an enzyme, which is known as Caspase. Normally, these are inactive. But whenever the cell want to do suicide, want to commit suicide, at that time, the cell will activate Caspase inside it. The cell will activate Caspase inside it. The cell will activate the Caspase inside it. Caspase are the enzyme that will degrade the cell. Into multiple small apoptotic bodies. You know, what is the apoptotic body? You will say, each apoptotic body contain a small amount of cytoplasm of this cell and small amount of nucleus of this cell. So small amount of cytoplasm and small amount of nucleus is present in each apoptotic body. The cell is entirely converted into multiple small cells. These are apoptotic bodies. Okay, during this entire process, there is no leakage. There is no leakage of cytoplasm. It's not bursting. There is no leakage of outside. And since there is no leakage, there is no inflammation. Neutrophils and inflammatory cells come whenever there is a leakage. So they come to know that something is happening. But during the entire process, it's silently. Silently the cell is converting into multiple small apoptotic bodies. There is no leakage outside. Since there is no leakage, there is no inflammation. In the end, a phagocyte will come. A phagocyte will come and engulf. It will eat or engulf all the apoptotic bodies. In this way, this cell is disappeared from the world. It is disappeared from human body, and that is apoptosis. How many of you got it? So come on, I will repeat it. Don't worry. I will repeat it multiple times. You will understand it. At the end, give me some time. Okay, so what is apoptosis? You tell me. First tell what is apoptosis. Say, ma'am, it's a type of cell death. Actually, there are two types of cell death, apoptosis and necrosis. Apoptosis is one of them. The first thing. It is a type of cell death in which the cell which is destined to die, it will activate an enzyme. The name of the enzyme is Caspase. The name of the enzyme is Caspase. Caspase will convert the cell, degrade the cell into multiple apoptotic bodies. In the end, a phagocyte will come and engulf all the apoptotic bodies. But during entire process, there is no leakage and no inflammation. Now, why I'm insisting the word no leakage and no inflammation? I'm again and again saying during the entire process, cytoplasm don't leak out. And since cytoplasm don't leak out, there is no inflammation. These are the 2 points which differentiate it from necrosis, the other type of the cell death. I told you, there are 2 types of cell death, apoptosis and necrosis. In apoptosis, there is no leakage, no inflammation. But in necrosis, there is leakage, there is inflammation. That's why I am saying apoptosis is suicide and necrosis is murder. You tell me, if someone want to commit suicide, what the person will do? Person will do it silently. Chup chap karega in a closed room, or in a person will not take a loudspeaker and a mic and announce I'm everywhere, I'm committing suicide. So it is silently. Silently means there is no leakage and no inflammation. That's why I am saying apoptosis is a suicide. But what if someone is getting murdered? The person will shout, oh, save me, save me. He will call everyone to save. So necrosis is the murder in which the cell will burst and the cytoplasm leaks everywhere. It leaks everywhere and it will cause inflammation. So this is apoptosis versus necrosis. Got it? As I told you. Everyone got it? Give me a thumbs up if you got it. Give me a thumbs up in the chat box. Got it? This is the definition. So what is the definition of apoptosis? Rather than learning this definition, I will look at the diagram. So this is a cell which want to commit the suicide. Now, when the cell want to commit the suicide, it activates an enzyme which is known as caspase in the cytoplasm. Caspase converts the cell into multiple apoptotic bodies. It degrades the cell into multiple apoptotic bodies. But during the entire process, there is no leakage, no inflammation. You have to highlight it. And in the end, a phagocyte is coming and engulfing all of them. This is the definition of apoptosis and we all got it. Okay, we all got it. Can we go ahead? Can we go ahead? You got it? Kamar Surya, you got it now? So this is the definition. Till now, I told you only definition. We will revise it again in the end. Now come on the mechanism of apoptosis on which you get many questions. You can make a 5-star over this topic. You get many questions over this. The mechanism of apoptosis. There are 2 pathways in apoptosis. I will explain you both. Extrinsic pathway, intrinsic pathway. You may be thinking, ma'am, what is extrinsic? What is intrinsic? Extrinsic means outside and intrinsic means inside. Now this is a cell. This cell wants to commit the suicide. This cell wants to commit the suicide. Okay. Now the signal, it will commit suicide only when it receives the signal that you are no more required, you commit suicide. So if the signal is coming from outside, it is known as extrinsic pathway. Extrinsic pathway. And if the signal is coming from our own nucleus inside the cell only, the nucleus only is giving the signal that I am damaged, you commit suicide. So the signal is coming from inside. Inside, then it is known as intrinsic pathway. So extrinsic and intrinsic is the difference, that from where the signal is coming. Signal is coming from outside, it's extrinsic pathway. Signal is coming from inside the cell, it's intrinsic pathway. So there are 2 pathways. First, understand the meaning. Now in each pathway, there are two phases. Initiation phase, execution phase, initiation phase, execution phase. Okay. So what is happening now? Here in initiation phase, the signal will come from outside. Here in initiation phase, the signal will come from inside. So first, I will tell you, I will start with extrinsic pathway. And I will tell you the details of the mechanism. I will tell you the initiation of extrinsic. Then I will tell you the initiation of intrinsic, because the execution of both of them is common. So I'm going to tell you 3 movie stories now. You can understand it with the help of stories, interesting stories. The first story will be initiation of extrinsic. The second, I will do intermission. Then the second story is the initiation of intrinsic. Then I will do intermission. Then I will combine the 2 intermission together and tell you a third story. That is the execution of both of them. How many of you got it? How many of you got it? Can I start? Can I start? So this topic is difficult, but I will make a cakewalk for you. Let's start with extrinsic pathway, and let's start the initiation of extrinsic pathway where the signal will come from outside. That's why it's extrinsic. So let me start. Hana. So let's start with extrinsic pathway, the initiation of the extrinsic pathway. Okay, can you see a cell? This can be any cell in human body. Okay, this is a cell. On the surface of the cell, we have death receptors. These are known as death receptors. They are inactive, but the cells have it. These are known as death receptor. You will say, ma'am, what are death receptor? There are two types of death receptor. Fast receptor, which is known as CD95, या फिर TNF, tumor necrotic factor receptor, TNF. These are known as death receptor. Normally, they are inactive. Can you see a cell? On the surface of the cell, death receptor are present. It can be fast, it can be TNF. It can be any of the two, but they are death receptor. There are two types of death receptor, I told you. What are the two types of death receptor? Fast or TNF. Fast is more important. Fast, these all are death receptor. You can see, these all are death receptor. They are either fast or TNF. Fast and CD95 is one and the same thing. Okay, it's one and the same thing. Learn two names of fast. You get question on fast also, CD95 also. And second is TNF. So these are inactive because they are present at a distance from each other. This is a normal cell in my body functioning normally. Now, this cell has done its function in human body. Okay, and now it is no more required. So this cell will receive a signal. This cell will receive a signal in the form of the ligand. The ligand is coming and giving the signal. So see, the signal is coming from outside, not from inside. The signal is coming from outside in the form of the ligand, and that's why it's extrinsic pathway. I'm proving that it's extrinsic because the signal is coming from outside. So this, let me zoom this diagram. Can I zoom it? See, this is the zoom version. So this is the ligand. Ligand is the signal. It is the fast signal, fast ligand. It is a signal. It is coming from outside and binding with the fast receptor. So once the fast ligand bind with the fast receptor, as soon as the fast ligand bind with the fast receptor get activated. Till now, this fast receptor was inactive. Till now, these receptors were inactive. But as soon as they receive the signal from outside, you can see it is receiving the signal from outside. As soon as the fast ligand is coming, binding with the fast receptor, multiple fast receptor come closer. They see. Multiple fast receptor come closer. Till now they were at a distance. See, appreciate the distance between them. But as soon as they got the signal via ligand, multiple fast receptor come closer. Can you see? Multiple fast receptor come closer and they form a domain. On the cytoplasmic side. This is known as fast associated death domain. FADD. What does it known as? Say, what is the full form of FADD? Fast associated death domain. It is known by this name only. It is FADD. FADD is formed. Now, in the cytoplasm of this cell, we have caspase, but they are inactive. Caspase 1, caspase 2, caspase 3, 4, 5, 6, multiple caspases are present. But all these caspases are inactive. But because of the formation of FADD, fast associated death domain, the first caspase which is activated here, caspase number 8. So, FADD convert pro-caspase into active caspase. So, caspase 8, or you can say 10 also. 8 and 10 are the first caspase to be activated in extrinsic pathway. It's a very important PYQ, repeated multiple times. Initially, it was only caspase 8, but in the recent Robbins, they have mentioned caspase 10 also. So you can learn the two caspase 8 and 10. Caspase 8 and 10 are the first caspase, which is the first caspase which are activated in extrinsic pathway. So this is the same story. Whatever I have drawn, the line diagram, the same diagram. So I think this diagram will have a better understanding. You can see this also. So in this diagram, what you can appreciate, say, ma'am, I can see a cell membrane. This is the cell membrane of a cell. Appreciate the cell membrane. On the surface of the cell membrane, you can see this is fast receptor, death receptor. Normally they are at a distance from each other. But whenever the ligand is coming and binding, multiple fast receptors are coming closer. Since they are coming closer, they are making a domain. The name of the domain is FADD, fast associated death domain. And because of which, it activates the pro-caspase 8 into active caspase 8. It activates. Okay, you will see, ma'am, what's next? So you can understand with the help of flowchart. Don't read paragraph, don't write paragraph. Understand with the help of flowchart. So what's happening? Say, ma'am, there are fast receptors, death receptor on the surface of the cell. Whenever the ligand is coming, signal is coming, multiple fast receptor come closer and they form a domain, that is FADD. Since the FADD is formed, it activates two caspase, pro-caspase 8 and 10 into active caspase 8 and 10. So you will say, ma'am, what's next? This is the intermission, है ना? This is the intermission. After intermission, I will tell you the story later on. So let's hold the intermission at activation of Caspase 8 and 10. इसके आगे क्या होगा? What's next? I will continue the story in execution, but later on. Before that, I want to tell you one more story, है ना? That is initiation of the intrinsic pathway, and then here I will end the story somewhere, and then in the execution I will compile the two stories together and take ahead. How many of you got it? How many of you got it? Can we go ahead? You got it? Pratiksha, Pankaj, Medico's learning, Siddharth, Kamar. Everyone, can we continue? So chalo. So this is all about the initiation in the extrinsic pathway, है ना? Just make a hold that we are ending the story at activation of Caspase 8 and 10. What's ahead, I will tell you later on. मैं बाद में बता दूँ. After 5 minutes, I will tell you in execution pathway. Before that, I want to teach you the initiation of the intrinsic. Can I start the initiation of the intrinsic? Let's start intrinsic pathway. Here in extrinsic pathway, the signal was coming from outside in the form of the ligand. Here in intrinsic pathway, the signal will come from inside. That's why it's known as intrinsic, inside. So let me show you the intrinsic pathway, the initiation of the intrinsic pathway. Can you see a cell? I can see a beautiful cell in front of me. The cell is having a nucleus, and inside it, it is having a double-stranded DNA. Okay, all human cell. This can be any cell of my body from head to toe. Okay, this is a normal functioning cell. It is having a normal nucleus. All the cells have mitochondria also. You can see this is the mitochondria. You know, mitochondria have two membranes. Do you know that? The inner membrane and the outer membrane. The mitochondria have two membranes. Between the two membranes, there is a molecule which is known as cytochrome C. It is present between the two membranes only. It don't come out. If the cytochrome C will come out, now it will cause apoptosis. So you may be thinking that, ma'am, who is holding the cytochrome C inside? Because let me zoom this portion. Let me zoom this portion. Because, you know, in the mitochondria, we have an exit in the mitochondria. The door, the transit. It is known as mitochondrial permeability. Mitochondrial permeability. Mitochondrial permeability means the exit, the door, the transit. So you may be thinking, ma'am, why not this cytochrome C just come out of the door? Who is holding it? There should be someone to hold it. Now, otherwise it should leak out. So it is not leaking out. Normally, it do not leak out. This cytochrome C do not leak out because on the door we have put 3 guards. These are the 3 guards. These are the 3 guards having a danda in their hand. You go inside. I will not allow you to come outside. It's like that. You can learn like a story. So who are the 3 guards? BCL2, BCL-X and MCL1. So these are the 3 guards who are holding cytochrome C inside the mitochondria. They do not allow cytochrome C to come out of the transit. That's why I call these 3 guards as anti-apoptotic proteins. These are the anti-apoptotic proteins which are formed by anti-apoptotic genes. So these are the 3 guards which are sitting on the transit, on the permeability of the mitochondria, and they are holding cytochrome C inside the mitochondria only. They do not allow cytochrome C to leak out. So normally everything is going smooth. In all my cells, this is happening. This is functionally. So cytochrome C do not leak out because the DNA is healthy. The cell is functional. Now imagine if due to any reason, maybe due to physical, chemical or biological, due to any reason, the DNA get damaged. Due to some rays like UVIR, due to certain chemicals, pesticides or something, or due to some viruses, the biological, because of any agent, the DNA get damaged. So first P53 will try to repair it. We have a mechanic, it will try. But sometimes the damage is too much. It's beyond repair. So P53 is unable to repair it. Now this cell cannot survive. It is fixed because the nucleus is the main thing in the cell. The nucleus is damaged, the DNA is damaged, the cell cannot survive. We have to kill the cell. We have to ask the cell to commit suicide. You are no more required because your DNA is damaged. So here the DNA will give the signal to the mitochondria that I'm gone. Please commit suicide. Please start the initiation. Please do the initiation of the suicide. Please commit suicide. Please start the process. I'm no more functional. I am done. P53 tried to repair me, but I am unable to get repaired. I am damaged beyond it. So see, the signal is coming from inside. The signal is coming from inside, not from outside. The signal is coming from inside only. Andar se hi aa raha hai. That's why it's intrinsic pathway I am proving. Now compare and contrast with the extrinsic pathway where the signal was coming from outside in the form of the ligand. The fast ligand. And here the signal is coming from inside. So this is intrinsic pathway. So here when the signal is coming from inside in intrinsic pathway, the signal is given to the mitochondria. So as soon as mitochondria receive the signal, okay, I have to commit the suicide. So what it will do? It will replace the duties of that guard. So it will replace these guards, these 3 guards with 4 another guards. 4 another guards. Initially we were having BCL2, BCL-X and MCL. You get many questions here. They are replaced by BARC, BIM and BAD. है ना, box bath, bim bath. So these are the guards which open the door and they allow the cytochrome C to go out, है ना. So these are anti-apoptotic proteins which were closing the door, and these are pro-apoptotic proteins which open the door. So can I say that on receiving the signal, mitochondria replace anti-apoptotic proteins with pro-apoptotic proteins? Anti-apoptotic proteins are the proteins which closes the door and keep cytochrome C inside only. They prevent apoptosis. But pro-apoptotic proteins are the one which open the door. By door, I mean the permeability, the transit on the mitochondria. They open the door so that cytochrome C can easily leak out and cause the apoptosis. So can I say on receiving the signal in the mitochondria, the anti-apoptotic proteins are replaced by pro-apoptotic proteins? Yes, you have to learn the examples also. How many anti-apoptotic proteins you know? Huh? How many anti-apoptotic proteins you know? There are many examples. BCL2, BCL-X, MCL1 is few of them. And apart from that, pro-apoptotic, box, bath, bim, bath. These are. है ना? So we have replaced these 3 guards with another 4 guards. So what these guards will do? You will say, ma'am, they will open the door quickly. So as soon as we have changed the duties of the guards, these are the guards, they open the door. Once they open the door, cytochrome C will leak out. You can see cytochrome C is happily coming out. It is leaked out. Hana, cytochrome C now in cytoplasm. You will say, ma'am, what does it do? It is there in the cytoplasm. We can see it is coming outside, but what does it do in the cytoplasm? Now, I told you in the cytoplasm, we have many caspase. Caspase 1, caspase 2, caspase 3, caspase 4, caspase 5. We have many caspase. Hana. So basically, there is a caspase, caspase 9. One of the caspases, all the caspases are inactive. Hana. There is a caspase, caspase 9. Pro-caspase 9. Pro matlab its inactive version. It is combined. It is in conjugation with a protein known as APOF1. APOF1. They are the best friends. You can see they travel together. Who two together? Caspase 9. The inactive version of caspase 9. Pro-caspase 9 with APOF1. They are duo. है ना? They are dual. They always travel together. They are couple. है ना? APOF1 and pro-caspase 9. Now the rule is that you cannot make caspase 9 active unless and until it is combined with APOF1. If you want to activate caspase 9, you have to break the pair. You have to break the pair. You got my point? So caspase 9 will remain inactive unless and until it is combined with APOF1. So APOF1 is the guard of caspase 9. I will not make you active. You remain inactive unless until I am there. Hana, if you want to activate caspase 9, you have to break the pair. So what does the cytochrome C is doing? Cytochrome C, after leaking, Hana, it is leaking out. After leaking in the cytoplasm, it is asking APOF1, you come with me. I will be a better friend. You come with me. You leave caspase 9 and you make a pair with me. है ना? So उसको पटा के अपने साथ ले गया. If you say simply. So basically, cytochrome C take APOF1 with him. है ना? Cytochrome C take APOF1 with it. So caspase 9 ultimately get activated because of this. So basically, cytochrome C is activating caspase 9 with the help of APOF1. With the help of APOF1, pro-caspase 9 converted into active caspase 9. So caspase 9 is the first caspase which get activated in intrinsic pathway. What were the 2 caspases which are activated in extrinsic pathway? In extrinsic pathway, the first 2 caspases which get activated is caspase 8 and 10. And here the first caspase which get activated is caspase 9. You get questions on that. Learn the first was caspase in extrinsic pathway and intrinsic pathway. And learn the mechanism in detail. Got my point? Say yes. So what are anti-apoptotic proteins? And what are pro-apoptotic proteins? Anti-apoptotic proteins are the one which closes the door, which closes the transit in the mitochondria, and they prevent the leakage of cytochrome C. That's why prevent apoptosis. And pro-apoptotic proteins are the one which opens the door so that cytochrome C can leak out and causes apoptosis. The best examples of anti-apoptotic proteins is BCL2, BCL-X, XL actually, and MCL1, and flip is also there. Hana, these are the guards which closes the door, and these are the guards which open the door. I told you box, bim, bath, bark, and BCL-XS. Hana, so these are the main example. You usually get confused in BCL. See, BCL-XL is anti-apoptotic and BCL-XS is pro-apoptotic. Highlight that. Others are clear in your mind. There is no confusion in that. I hope. Okay. Can we go ahead? Okay. So I'm coming on your questions, Said. Wait, let me finish it. I will come on all your doubts, okay? I will give you a time to ask the doubts. Let me finish it. I'm in the middle of something, okay? So this is the intrinsic pathway, initiation of intrinsic pathway. So here the signal is the DNA damage. When the DNA is damaged beyond repair, it cannot be repaired. Hai? So anti-apoptotic proteins are replaced by pro-apoptotic proteins in the mitochondria, in the mitochondrial transit. I mean BCL2, BCL-X, MCL1. These are 3 anti-apoptotic proteins. And they are replaced by pro-apoptotic proteins like BARX, BART, BIM, and BAT. So they will open the door. They will increase mitochondrial permeability. They will open the door. Once they will open the door, cytochrome C will leak out. Once cytochrome C is coming out, it will take APOF1 with him. It will take APOP1. So pro-Caspase 9 is alone and pro-Caspase 9 converted into active Caspase 9. So this is the story. This is the flow chart. In this way, you have to understand, write or make the notes. Okay. So we are done, है ना, of extrinsic pathway ka initiation and intrinsic pathway ka initiation. What's next? So what was the story here? We ended the story at the activation of Caspase number 8 and 10. And here we ended the story at the activation of Caspase number 9. You'll say, ma'am, what's ahead? Aage kya? So what's ahead here and what's ahead here? We will continue the story in execution, which is common for both of them. So execution is the convergence point. It's also a question. Execution for both the pathways convergence. It's common. So if you're talking about the intrinsic, so it's a convergence point. The execution is the convergence. If you're talking about extrinsic pathway, the first caspase activated is 8 and 10. You know the mechanism via death receptor. So multiple fast receptor coming together, they form a domain FADD, and FADD causes the activation of 8 and 10. If it is an intrinsic pathway, the first caspase which get activated is caspase 9 with the help of anti-apoptotic proteins replaced by pro-apoptotic proteins, है ना, and APOP1. ठीक है, caspase 9 is get activated. Now next, caspase number 3, 6, 7 get activated. So here caspase 8 and 10 and here caspase 9. They activate the next caspase 6, 3, 6 and 7 and ultimately all the caspases are activated. Once all the caspases inside the cell are activated, है ना, they degrade the cell. They degrade the cell into multiple small apoptotic bodies. I told you each apoptotic body contain a small portion of the cytoplasm and a small portion of the nucleus. The nucleus also get fragmented. So during the entire process, there is no leakage. Since there is no leakage, there is no inflammation. Neutrophil do not come to know that anything is happening outside. It's silently, like suicide. If someone is committing suicide, it's silent. So in the same way, it's also silent. है ना? All the caspases get activated. They will degrade the cell into multiple apoptotic bodies. Got my point? So here 3 questions are there. The first caspase which get activated in extrinsic pathway ka initiation, say caspase 8 and 10. The first caspase which get activated in intrinsic pathway ka initiation, say caspase 9. And the first caspase which get activated in execution. Execution is common for both of them. So answer is 3, 6, 7. So these are the questions which are usually asked, है ना? These are the questions which are usually asked in your exam. So take care of them. Got my point? So ultimately multiple apoptotic bodies are formed. Okay. So definitely, Satyaben, I'm going to continue with all the series with on the YouTube which are incomplete. I'm going to complete. That's why we have started these live session. So don't worry, stay connected and you can write down the exact topics which you want by my side on the YouTube. Okay. So let me come on the next. Okay. Now can you see this diagram? It is a compilation of the two. It is a compilation of extrinsic as well as intrinsic. So inside a cell, it can be extrinsic, it can be intrinsic pathway, and ultimately the execution is common for both of them. And also you can understand. Now coming on the morphological changes in the apoptosis. Now this is the mechanism of the apoptosis. Anna, you want me to summarize the mechanism of apoptosis? In the mechanism of apoptosis, we have 2 pathways. Extrinsic pathway, in which the signal come outside, or intrinsic pathway in which the signal come from inside only. So in extrinsic pathway, when the fast ligand come, it bind with fast receptor. है ना? So multiple fast receptor come closer and they form a domain that is known as FADD. And this FADD cause the activation of caspase 8 and 10. This is the story over here. In intrinsic pathway, whenever there is a DNA damage, the signal come from inside only. The nucleus will give signal to the mitochondria. In the mitochondria, anti-apoptotic proteins, that is BCL2, BCL-XL, they are replaced by pro-apoptotic proteins, that is Box, Bath, Bim, Bath. है ना? So once this happens, cytochrome C will leak out. Cytochrome C, after coming out, it will take APOP1 with him. That is apoptotic activating factor. So caspase 9 become alone. So caspase 9 is the first caspase which get activated here. So here we have caspase 8 and 10. Here we have caspase 9. So the next caspase, they will activate. Execution is common. They both of them together activate caspase number 3, 6 and 7. And ultimately all the caspases get activated. And ultimately the entire cell is degraded into multiple apoptotic bodies. And in the end, a phagocyte will come and engulf all of them. Can we go ahead? Say yes if you got it. Can we go ahead? Okay, definitely say it. I will do so, definitely. Anyone having any doubt in this apoptosis mechanism? Otherwise, give me a thumbs up if you got it. The complete mechanism is in front of you. The summary I have given you. So in apoptosis, the mechanism, we are having two pathways, the extrinsic and intrinsic. Now, I am going to tell you the morphological changes in apoptosis. Now, whether it is extrinsic pathway or intrinsic pathway, this is common for both. So morphological changes in apoptosis. I'm going to tell you the morphological changes in apoptosis. Can you see this is a normal cell? This cell is normal cell. Now this cell is getting apoptosis. Either by extrinsic pathway or by intrinsic pathway. The mechanism is different, but ultimately morphology is same. So the first thing which is going to happen, it's common for extrinsic, intrinsic both. The first thing which is going to happen is cell shrinkage. 6 things happen one by one. You have to learn the sequence. The first thing is cell shrinkage. The cell will get shrink. So you get a question, the earliest change in apoptosis, answer is cell shrinkage. The answer is cell shrinkage. Okay, the earliest change in apoptosis, the answer is cell shrinkage. This cell gets shrinked. This cell gets shrinked. Now inside the cell we have cell organelles. We have mitochondria, we have ribosomes, we have endoplasmic reticulum. When the cell gets shrink, the multiple cell organelles, they come closer, they become compact. That's why the cytoplasm of the cell look more pink in color. So the second thing which is happening is eosinophilia. Actually shrinkage leads to eosinophilia, you can understand like that. So the earliest change is shrinkage, and because of the shrinkage, multiple cell organelles, they come closer, they become compact. So cytoplasm look more pink. Pinkishness in the cytoplasm is eosinophilia. So the second thing happening is eosinophilia. The third thing is the nucleus changes. See the nucleus here? It's dark blue compact. It will become more compact. Condensation of the nucleus, compactness of the nucleus. So it is known as pyknosis. Pyknosis means it become more condensed, more blue, more condensed, more shrinked. Pyknosis. And after pyknosis, the nucleus become fragmented. The fragmentation of the nucleus is known as Karyorrhexis. So two things happen in nucleus one by one. First, the nucleus become condensed. Condensation of the nucleus is known as pyknosis, and then the nucleus get fragmented. Fragmentation of the nucleus is known as Karyorrhexis. So this is the third change, that is nuclear change. After that, in the cell membrane, multiple small blabs are formed. Can you see? Multiple small blabs are formed. So the fourth thing is the blab formation. First, there is cell shrinkage, you can see in the diagram. Then there is eosinophilia, number second, see in the diagram. Third is the nuclear changes, that is pyknosis and Karyorrhexis. I hope you understand what is pyknosis, what is Karyorrhexis. Okay, the fourth thing is the blab formation. On the surface of the cell, multiple small blabs are formed. Okay, now each blab, see, each blab, this is a blab. It will carry a small portion of cytoplasm and a small portion of nucleus. The nucleus is already fragmented with it and separate from the cell. So all the blabs will separate from the main cell. So basically the cell is converted into multiple small apoptotic bodies. In this way, the apoptotic bodies are formed. You got it? So all the blabs get separated from the main cell, carrying a small amount of cytoplasm and nucleus with them. So complete cell get converted into multiple apoptotic bodies. But during the entire process, there is no leakage. The cytoplasm do not leak out. And since there is no leakage, no inflammation. In the end, what's happening? A phagocyte is coming. In the end, a phagocyte is coming and engulfing all the apoptotic bodies. It's engulfing all the apoptotic bodies. The cell disappeared from the world. Give me a thumbs up. Give me a thumbs up. Everyone got it? So currently you can ask the questions which is related to the apoptosis. Whatever topic you find difficult, you can message me personally instead of writing down in the chat box and it get distracted everyone. So whatever, Shubham, definitely I will take the auto guides. But if any topic you find that I should take it on the YouTube, you can message me. I will try to schedule it as soon as possible. Okay. But currently if you have any doubt related to the apoptosis, please ask it. You are open to ask. Otherwise, give me a thumbs up. Can we go ahead? Can we go ahead? So these are the 6 changes. Can you enumerate? Close your eyes and enumerate the 6 in sequence. 1, 2, 3, 4, 5, 6. Tell me the 6 morphological changes takes place in apoptosis. What is the earliest? The first is cell shrinkage. Say in a sequence only. The sequence is important. The first is cell shrinkage. Okay, what is the second one? Due to the cell shrinkage, there is eosinophilia. Okay, there is eosinophilia. The second is. So there is cell shrinkage. There is eosinophilia after that. After that, the third is the nuclear changes. That is pyknosis and Karyorrhexis. Karyorrhexis. That is fragmentation of the nucleus. Condensation and fragmentation of the nucleus. Then multiple blabs are formed. Now each blab will carry a small amount of cytoplasm and nucleus and separate. So multiple apoptotic bodies are formed. Apoptotic bodies are formed. And in the end, a phagocyte will come. And engulf all the apoptotic bodies, that is phagocytosis. Now 3 questions are asked over here. Let me ask you 3 questions and highlight 3 questions. Earliest change in apoptosis. I guess everyone know. The earliest is the cell shrinkage. earliest, है ना, last stage of apoptosis, all apoptosis, whether extrinsic, intrinsic, the last, it ends with the phagocytosis. So last stage in the apoptosis, the morphological change is the phagocytosis. Everyone know that. But the third question, most specific or characteristic feature of apoptosis. Most specific is which one, or characteristic? So answer is the nuclear changes. So nuclear changes is the most characteristic or specific feature of apoptosis. So learn the three questions very clearly in your mind. The earliest morphological feature, the last morphological feature, and most specific or characteristic morphological feature. How many of you got it? How many of you got it? So please learn these three questions very clearly, and you should be able to enumerate as well as draw as well as understand. Try to draw maximum diagrams so that it fit in your permanent memory. Now coming on the diagnosis of apoptosis. For example, I'm giving you a HNA slide of a tissue. It can have any tissue. So inside that tissue, you can see many cells are there. This is a biopsy, for example. It is having many cells inside that. It is having thousands of cells. I want you to highlight which is apoptotic cell. So you will say, ma'am, apoptotic cell will be smaller as compared to others. It is more pink as compared, but sometimes we have difficulty to identify them. Out of all the cells, thousands of cells, which cell is apoptotic cell? So is there is a way to do the diagnosis of apoptosis? There are two ways to do the diagnosis of apoptosis. What are the two ways of diagnosis of apoptosis? The first is the apoptotic marker. The name of the apoptotic marker is Annexin 5. Annexin 5. Now, I told you this is a slide on which there is a biopsy or there is a tissue. And it is having many cells, thousands of cells on that. So, I want to highlight the apoptotic cell. So, there is a marker which is known as Annexin 5. And it's available commercially. Annexin 5. I will spread Annexin 5 over it completely. Leave it for a few minutes and then wash it. So, Annexin 5 will highlight only apoptotic cell, not other cell. Annexin 5 will highlight only apoptotic cell, not other cell. You will say, ma'am, okay, so this cell, so you can see the slide under microscope. You will understand which is apoptotic cell. So whatever cell is marked by Annexin 5, that is apoptotic cell. It's a rule. You will say, ma'am, why? What is the principle behind that? Why Annexin 5 is highlighting only apoptotic cell, not other cell? So you have to understand it. So imagine this is a normal cell, the cell membrane of a normal cell. This is a normal cell, and this is converted into blebs. I mean, this is an apoptotic cell. You see the blebs are formed. This is apoptotic cell. Apoptotic cell. Okay. On the normal cell, there is a phospholipid on the inner side of the cell membrane. The name of that phospholipid is phosphatidylserine. Phosphatidylserine present on the inner side of the cell membrane in all our normal cells. But whenever any cell do apoptosis, so basically there is flip-flap, ulta-pulti, flip-flap, Hindi mein ulta-pulti, flip-flap of the cell membrane. The cell membrane does flip-flap. When the cell membrane do flip-flap, so phosphatidylserine, instead of inner side, it will come on the outer side. So phosphatidylserine comes on outer side. This is known as externalization of phosphatidylserine. So we can say during apoptosis, the phosphatidylserine undergo externalization due to flip-flap of the cell membrane. Now, you put Annexin 5. Annexin 5 over here also. And Annexin 5 over here also. Annexin 5 bind with phosphatidylserine. So you yourself tell, will it bind with this cell? No. Here phosphatidylserine is on inner side. Will it bind with this cell? Yes, it will bind because phosphatidylserine is on outer side. So it will highlight only apoptotic cell, not normal cell. This is the reason you get questions on that. Phosphatidylserine is the principle of Annexin 5. Yes, Pratiksha, yes. But phosphatidylserine is the principle. So please understand. So it is the phosphatidylserine. Due to phosphatidylserine, the externalization of phosphatidylserine. How many of you got it? Now in the diagram also, the same thing is shown to you. I guess you can see this is a cell membrane and you can see this is also a cell membrane. Okay, this is normal cell, this is apoptotic cell. Okay, can you see it? Now you can see here phosphatidylserine is present on the inner side of the cell membrane, and here phosphatidylserine is present on the outer side of the cell membrane. And whenever you are putting Annexin 5, so it is binding here but not here. So this is the principle behind Annexin 5. Okay, now the second method is the electrophoresis. I told you again, see this is a normal cell and this is the nucleus of the normal cell, and this is the apoptotic cell in which nucleus undergo fragmentation, that is Karyorrhexis. So in apoptotic cell, this is normal cell and this is apoptotic cell. Apoptotic cell. In apoptotic cell, the fragmentation of the nucleus takes place with the help of enzyme which is known as endonuclease. Endonuclease is the enzyme. On the endonuclease, it is doing the fragmentation, you can see. Now do the electrophoresis. Do the electrophoresis of this cell as well as this cell. Agarose gel electrophoresis. So here you will get the DNA in a single length, the complete DNA. But here the DNA is fragmented with the help of endonuclease into multiple small bodies, multiple small nucleus. That is Karyorrhexis. So you will get the DNA in multiple fragments. So here the DNA is in a single length in electrophoresis. But here in electrophoresis, you will get this pattern. So looking at the electrophoresis pattern, what is this pattern known as? It is looking like a step ladder. CD ke jaisa, step ladder. So this is known as step ladder electrophoresis. Step ladder pattern in the electrophoresis. So step ladder pattern in the electrophoresis indicate that this cell is an apoptotic cell. How many of you got it? Now look at the apoptosis. Look at the electrophoresis. Can you appreciate this step ladder? So this is an apoptotic cell. But here it is more or less, it's the same. I mean continuous. Here also it's continuous. But this is the apoptotic cell. So in apoptotic cell, on agarose gel electrophoresis, we get step ladder pattern due to the cleavage of the DNA by endonuclease. Say yes. Got it. Got it. Can we go ahead? Huh? Can we go ahead? So everyone got it. Does anyone have any doubt till here? Huh? So we are done with the apoptosis. I told you the mechanism, I told you the morphological changes, and I told you the diagnosis. Now I'm telling you the differences between apoptosis and necrosis. So I'm teaching you cell death. I told you there are two types of cell death. One is apoptosis and one is necrosis. In both of them, ultimately the cell is dying. But this is like suicide and this is like murder. In suicide also the person dies. In murder also the person dies. So cell is dying in both of them. But the mechanism is different. Why I'm calling it suicide and why I'm calling it murder? You know, suicide is done by one person. It is not the complete city is doing suicide. The complete village is doing suicide. No, one person. So apoptosis takes place in single cell. Now you can correlate. But murder can be done of a multiple person at one time. The multiple persons, a group can be murdered together. So here it takes place of a group of cell. So apoptosis is always of a single cell and necrosis is always a group of cell. First thing. In apoptosis, the cells shrink. That's why the cell membrane is intact. There is a cell membrane is intact. The cell is shrinking. Shrinking. So cell membrane is intact. No leakage, no inflammation. But in necrosis, the cell will swell. See the contrast. Here the cell is shrinking. Here the cell is swelling. Since the cell is swelling, here the cell is swelling, it will burst. So it will burst out. Since it will burst out, there is leakage, there is inflammation. So whenever there is a suicide, it's silently. No one come to know. It is in a closed room. It is silent. That's why no leakage, no inflammation. But whenever someone is murdered, it will shout and save, and call everyone. So here leakage is present, inflammation is present. So here leakage, inflammation absent. Here leakage, inflammation present. Here it is of a single cell. Here it is of a group of cell. Here the cell is shrinking. That's why the cell membrane is intact. And here the cell is swelling. That's why the cell membrane is bursting. Okay, got it. Can we go ahead? Huh? Okay. Thank you, Jai Kumar. Thank you for the compliment. You can see the two diagrams. You can understand apoptosis and necrosis here in the diagram. Can you see this is apoptosis? Can you see necrosis? The same thing which I taught you here, you can see it here. See this cell? I want you to highlight this cell. Okay. Can you see this cell? This is a normal cell. Can you see this cell? This is a normal cell. So here the suicide will take place and here the murder will take place. You can understand. So here the cell is shrinking. Appreciate the size here. And here the cell is swelling. Appreciate the size here. You can yourself appreciate. Here it's shrinking and here it's swelling. The first thing. Here since it's shrinking, it is converting into multiple apoptotic bodies. All the apoptotic bodies separate from the main cell, forming blebs. And in the end, a phagocyte will come and engulf all of them. A phagocyte is coming and engulfing all of them. No leakage, no inflammation. But here the cell is bursting out. It's bursting out, everything leaks out. So it is causing the neutrophils. Leakage is there, inflammation is there. I hope you got it. Now, if you are preparing for university exam for a second prof, these are the list of important questions from this topic and I'm damn sure you can write down on any. You usually get a long question on apoptosis or you get a small one of the mechanism, either extrinsic pathway or intrinsic pathway or morphological or diagnosis, one of the part as short question. So today in apoptosis, we have learned what is the definition of apoptosis? You all can define with a diagram. What are the two mechanisms? Extrinsic pathway, intrinsic pathway. In each of them, two phases, initiation, execution. We can draw the flow charts and diagrams. You know the diagnosis, the two ways of diagnosis of apoptosis, Annexin V and electrophoresis. And you know the differences from necrosis. You know the five differences between them. So you can easily understand it. Now, if you're preparing for any of the competitive exam, we will solve all the MCQs based on that in the previous year questions of the medical entrance competitive exam. The first question is in front of you. Can you give the answer in the chat box? I want everyone to participate. Please write down your answer in the chat box. CD95 is a marker of intrinsic pathway of apoptosis or extrinsic pathway of apoptosis or monocyte or leukocyte. Leukocyte is WBC. So the other name of CD95 is FAS. I'm giving you a clue. The other name of CD95 is FAS, I told you. Hai na? So it is involved in which pathway? Yes, it is involved in which pathway? I'm asking you. It is involved. Yes, absolutely right, Pankaj. What about others? Pankaj, yes, Pratiksha. So is it A or B? So CD95 FAS is required in extrinsic pathway, not intrinsic. Extrinsic pathway में death receptors थे ना? We started. Extrinsic pathway have two death receptors. So on the surface of the cell, we have either CD95 or FAS, or we have TNF. They are at a distance from each other. But whenever they get the ligand, they come closer. So it is the extrinsic pathway, not intrinsic. So correct answer is B, and you all are right. Very good. Very good. If I change the same question, currently the answer is B. Hana, if I change the same question to cytochrome C, what is your answer now? Cytochrome C is a marker of which pathway? Hana, or if I change the question, APOF1 is a marker of which pathway? Cytochrome C and APOF1 were involved in which pathway? So they are involved in intrinsic pathway. In intrinsic pathway, between the two layers of the mitochondria, cytochrome C is present. And once it leak out, it takes APOF1 with him. Hana, so cytochrome C or APOF1 pe bhi question aata hai. You get a question on cytochrome C. So in that case, your answer become A. I hope you got it. Everyone got it? Everyone got it? So in this way, the questions can be asked. The next question, I guess we have already done. In apoptosis, cytochrome C acts through what? It acts via APOF1, BCL2, FADD or TNF. So cytochrome C, after leaking out from the mitochondria in the cytoplasm, it takes APOF1 with it. So that the caspase 9 become alone. Hana? So the mechanism of action of cytochrome C is via APOF1. So I hope you all can understand. Absolutely right. The correct answer is A here. Hana? The next question is in front of you. Which of the following is an anti-apoptotic protein? Is it BARC, BAD, BCL-X or BIM? So you have to understand the two type of the proteins, the two type of the genes, the two type of the proteins, anti-apoptotic and pro-apoptotic. If you understand the intrinsic pathway, you can understand it well. Anti-apoptotic are the one which closes the door and they prevent the leakage. They do not allow the leakage of cytochrome C. Okay? But pro-apoptotic are one which open the door and they easily leak out the cytochrome C. They leak the cytochrome C. So what are the examples here? Here, the examples are BCL2, MCL1 and BCL-XL. Here we have, I'm sorry, BCL-XX. Apart from that, BARC, BAD, BIM and BARC. So these are the examples. Now read your question. They are asking anti-apoptotic. So which of the following is an example of this? So correct answer is BCL-X, better if they give XL. But if they are not giving, at least we will go with this. But BARC, BAD, BIM, these are definitely pro-apoptotic. I hope you can understand. Okay, the next question is in front of you. You can answer it. What is the correct answer? What is the earliest change in apoptosis? Obviously cell shrinkage. Obviously cell shrinkage. We all know. But if I change the question, instead of earliest, if I ask what is the most specific change or what is the most characteristic change in apoptosis, what is their answer now? What is their answer now? Earliest is cell shrinkage, we all know. What is the characteristic or most specific change? What is the answer in that case? Yes, in that case, the answer will be Pyknosis. Pyknosis and Karyorrhexis is the answer. That is, nuclear changes are more specific. Okay, the next question is the characteristic feature of apoptosis. Cell membrane remain intact, say yes or no. There is basophilia, say yes or no. Nuclear molding is there, say yes or no. And cell swelling is there, say yes or no. So only one of them is correct. The cell membrane remains intact. That's why no leakage, no inflammation. But it's not basophilia. Actually, it's eosinophilia. It's not nuclear molding. It's Pyknosis and Karyorrhexis. Okay. It's not cell swelling. It's cell shrinkage. So other three options are wrong. Only one is correct. That is A. Cell membrane remain intact till end. Rather, blab formation bhi ho jayega. Cell membrane will not disrupt. It's intact till end. The next question is in front of you. All of the following are the features of apoptosis except which of the following is not a feature of apoptosis? Cell swelling occur or do not occur? Nucleus compactness. Compactness of the nucleus matlab Pyknosis. It occur or do not occur? Cell membrane remain intact till end. Is it there or not there? Eosinophilia. Yes or no? So I guess eosinophilia is there. Cell membrane remain intact. That's why no leakage, no inflammation. Nucleus become compact, but it's not cell swelling. It's cell shrinkage. So A is wrong among them. So questions are very simple if you know the mechanism. Annexin 5 is a marker of. I guess this is the easiest ever question. The correct answer is apoptosis. The mechanism is through phosphatidylserine. During apoptosis, there is externalization of phosphatidylserine. That's why annexin 5 is positive in apoptotic cell, not in normal cell. Okay. Now, if you want the notes of this session, you can see the chat box right now. In the chat box, in the first, there is a pinned link. If you click on the link, you will get the notes PDF of all my YouTube session, which I'm taking in this live series. So in the last session also, this session also, and the next sessions also, one by one, we will all put all the notes there. So kindly, those who want the notes, please copy the link or click on the link. You will be diverted towards the notes. Now, thank you for your attention, for your precious time. If you have any doubts regarding patho, pharma, micro, PSM, or whatever subjects I'm teaching you or whatever topics I'm teaching you, so please text it on this number. It's 9321001485. You can note it down. This number is available only for the chat support, not for calling. So don't call, just do the chat support, whatever query you have related to, or you have some topics in your mind that I should be taking for your exam. These are the difficult topics and you want me on my side to take it on the YouTube. I will do that. And you can do a scan and you will be diverted towards my website, that's medlive by drpriyanka.com, and there you can connect with me on multiple social media platforms, that is Instagram, WhatsApp, Telegram and all. So all the links are given there and you can connect with me for further study material for your exams. Thank you so much. Thank you so much. Now, what about the next class? So this class is done. This is done. The next class is on necrosis. Don't dare to miss it. So every Monday, Wednesday and Friday at 9:30 a.m. in the morning, we will be live with every time new and important and difficult topics. So the next class we are having on the Wednesday. On the Wednesday, that is 6th of December. Today is Monday. A day after tomorrow on Wednesday, we will be live with necrosis, the ultra important topic. We will be doing all the questions related to that. Okay. So thank you so much. Thank you so much for being with me and giving your precious time to me. Okay, just a second. Okay, thank you so much. I would like to end it. Okay, bye-bye. Thank you so much. Yes, I will schedule it definitely. Definitely, I will schedule all these topics one by one. Thank you so much for your compliments. Thank you so much. I'm ending it. Bye-bye. Wishing you all the best.